Okay, so again, hello everyone. Thank you for joining us today. Whether you work in the industry or academia, you know that biotech R&D can often be fragmented, expensive, and slow. But what if software could change that? So that's exactly what we're here to talk about today. So first, I'll begin introducing the speakers for today. So on one hand, we have Eduardo Abeliuk. Say hi. He's an entrepreneur and scientist with a PhD in electrical engineering and a master's degree in bioengineering from Stanford University. He's also one of the co-founders of Teselagen, where he leads the company's strategic direction. Eduardo has a deep passion for using technology to streamline biological R&D. And on the other hand, we have Álvaro Olivera Nava. You can wave, say hi. He's a chemical engineer with a PhD in chemical engineering and a deep knowledge and passion for synthetic biology. He has held research positions in Europe and Latin America. And at Teselagen, Alvaro focuses on developing cutting-edge tools for automating biological design and accelerating biotech discoveries. And also here we have Minerva Castellano. You can also say hi, Mine. And she will be reading and helping answer the questions that you can share in the QA section down below. And finally, I'm Jota Hurtado, and I'll be your host for today. So, what to expect during this webinar? Eduardo will start with a brief high-level overview of the Teselagen's platform. Then Alvaro will introduce the fundamentals of sequence alignment and PCR primer design in Teselagen. He will then provide a brief live demo of the alignment tools and automated primer designers, illustrating how these tools streamline sequence comparison and verification, PCR setup, and overall experimental planning. And finally, we'll open the floor for Q&A at the end of the webinar. So throughout the webinar, feel free to write your questions in the QA section at any time. I mean, I will also be looking at them. We will address as many as possible during the QA session at the end of the webinar. And questions we are unable to answer today, it will be followed via emails in the coming days to everyone that participated today. So without further ado, I'll hand it over to you, Eduardo, to kick things off.
Thanks, Jota. Welcome, everyone. So before diving into today's topic, I would like to start with a brief, very high-level overview of Teselagen, why we exist, and what our platform is all about. And as many of you know, developing biological products comes with significant challenges. There are underlying experiments on one hand, and processes tend to be fragmented, inefficient, and prone to error. And these limitations make it very hard to iterate quickly, to connect insights across workflows, and to learn effectively from prior work. And moreover, biological product design is by nature, we believe, an iterative process which requires the ability to systematically track and connect every stage of development from design through build to phenotypic testing so that data and learnings can flow seamlessly throughout the entire cycle. So that's why, exactly why at Teselagen, we've built the next generation platform for biological R&D. And it's one that is really reimagining how biological products are designed, built, and optimized. Our platform brings structure and intelligence to the entire development process, and it's organized into 4 powerful modules, each focused on a key phase of innovation. On one hand, on the left, biological product design, where ideas take shape, then experimental workflow design, where they are brought to life, then biological data analysis, where insights emerge, and finally, biological product optimization, where everything comes together to guide the next iteration. And altogether, these stages form what we all know as the design, build, test, learn cycle, or DBTL cycle, in which our mission as a company is to empower scientists like you to not only implement the DBTL cycle, but hopefully to close it, because that's where the real power lies, capturing the knowledge from each iteration, learning from what worked and what did not, and using those insights to guide smarter designs, better experiments, and ultimately better products. We're also very proud to have built this platform in close collaboration with a diverse network of scientists, with leading companies and institutions, ranging from startups to larger companies, and to national biofoundries, working across multiple biotechnology sectors. And along the way, we've also integrated with some of the leading technology providers in the field, creating a truly connected ecosystem for modern biological R&D. Our platform is divided into a number of toolkits that you see on the screen, that are unified under a common user interface. And some of these toolkits, now highlighted in blue, fall under our so-called design tools, and are useful for things like planning your experimental designs and tracking basic experiments through our electronic lab notebook. And we also have another set of tools and toolkits that are useful for then tracking physical inventory, planning and running experimental workflows, which are particularly useful when you need to be ready to scale up your experiments in terms of number of samples being tracked, throughput of experimental workflows being run, or simply many times automating certain processes within your workflows. And finally, we also have some toolkits for ingesting experimental datasets into our platform, parsing and organizing those datasets, performing statistical analysis, visualizing the datasets, and using the datasets to train more advanced AI models. Finally, our platform would not be complete without a series of powerful interoperability tools that allow it to connect to different third-party systems, such as lab equipment, different vendor APIs, and third-party databases, and also a number of third-party software tools. Importantly, we've been designing Teselagen in such a way that can, on one hand, be connected with third-party systems through our API in a number of ways, and it can provide both a good user experience to data scientists on one hand, who prefer having programmatic access to our platform, as well as many biologists who more often than not prefer simply accessing our tools and integrations directly through the user interface. For today, though, the focus will be placed on a few of these tools highlighted here in blue, namely, a few core entities from our molecules library and a few tools from our molecular biology toolkit. And these tools, including other ones for designing genetic constructs and primers and the alignment tools, together with our electronic lab notebook, are all included in our Teselagen Community Edition. This Community Edition is available for everyone for free. It has been designed to make these design tools accessible to everyone. Already over 300 institutions have recently signed up, and it's because it's cloud-based, you can easily access it and get instant upgrades regularly. And of course, we would love you to try it out and see what you can build. You can find more information directly from our website, where you can find a link to sign up for free, or you can go directly to community.teselagen.com to sign up. And with that, I will hand it over to Alvaro, who is going to walk you through some practical examples of how Teselagen brings some of these tools to life, especially how you can streamline primer design and sequence analysis. So Alvaro, the floor is all yours.
Thank you, Eduardo. And thank you all for being here with us today. Today I will be introducing two Teselagen design tools for molecular biology and for analysis and experimental design. We will talk about the sequence alignment tool and the PCR and primer design tools. We have talked about other different tools in previous webinars, especially the one Eduardo mentioned for DNA assembly. So today we will focus on sequence alignment first. And here I want to highlight what sequence alignment is used for in practice. And probably many of you know that we typically use it to compare variants or to verify cloned inserts or to check differences between a reference sequence and an experimental sample. The Teselagen alignment tool enables both pairwise and multiple sequence alignments, and we can clearly visualize mismatches and indels there. And we can also switch between nucleotide and protein views, depending on the level of detail required and the type of alignment we select. Particularly in protein alignments, we can also, we always look for information, and we look for conserved and non-conserved residues, and sometimes we can learn something about protein function by comparing different sequences. The key advantage is how fast we can go from raw sequences to a clear, interpretable alignment and exportable report in Teselagen. The automated PCR primer design tool, here we have another problem. We need to calculate and to design primers for any PCR we would like. So here, instead of manually calculating, let's say, melting temperatures or checking GC content, we just set high-level constraints and we specify details, such as, for instance, the desired amplicon size range or the primer length limits. Later, if we like, we can also include optional 5' modifications if needed, or we can modify the sequence of the primers even. But the system, Teselagen, then generates candidate primer pairs automatically for you, and you can select from a list of different primer pairs. In the next slide, after generating candidates, the platform performs several checks in the background. The same things you do manually usually, or the same thing you can use some other platforms to do. You check thermodynamic balances, you check GC content, you check for hairpins and formation of hairpins, self-dimers, cross-dimers, off-target binding, many different things. But in this case, the tool just gives us a list of ranked primer pairs, and all the calculations are done inside Teselagen. So you don't have to worry about that. So in the next slide, this is just the abstract of everything else. The main advantage of doing PCR design here in Teselagen is efficiency. You don't waste time. So we don't have to manually optimize details because that's cumbersome. So this reduces trial and error, this lowers the chance of failed PCR runs and saves time for you. Plus, everything is integrated with inventory tracking and workflow automation if you'd like to use it in Teselagen, and that makes it easier to scale and to document your work. So let me finish with just a quick reminder of best practices for PCR design. So you have to keep pair TMs within a tight range. You have to avoid 3' complementarity. You have to reserve 5' add-ons for adapters or tags as you want, and watch out for repetitive or palindromic regions. You have to calculate everything. The good thing is here, as I told you before, Teselagen automates everything for you. So you can focus on your experiments and not the setup. So now, instead of continuing talking, I will share my screen with you, and I will show quickly how to use these tools in Teselagen. So let's start with the sequence alignment tool. The best way, there are many ways you can do so in Teselagen, many ways of getting the same result, but the easiest way is to go directly to sequence alignments here in your left-hand menu in Teselagen. When you go to sequence alignment, you have a list of all the alignments you have performed in your system. You can, of course, review any of them. But if you want to create a new one, you just click here and you select if you want a nucleotide alignment or an amino acid alignment. Let's start today with a nucleotide alignment. And in this case, you can select if you'd like a multiple sequence alignment, a pairwise alignment, or if you have sequences, a reference sequence, and short sequences derived, from example, from sequencing reads to align to this reference sequence. So you can choose any of these three. Let's choose the multiple sequence alignment because that's the best things I can show you. When you choose that, you have, first thing, you have to select your sequences. You can upload them from a file, so any of these formats you can use. You can manually add them, just pasting your sequences here or just writing them, not usual, but you can do so. But the best way is to go to your Teselagen library and choose your sequences. So in this case, we will go to my DNA sequences here in Teselagen, and I want to align all my hemoglobin sequences in Teselagen. So I just filter for hemoglobin, select all of them, 23 of them. I have alpha hemoglobin sequences here coming from different organisms. So I choose those sequences. So first thing, the system detects that there are some duplicates. You can remove them and keep only representative sequences. But in this case, I want to compare all the sequences from all my organisms. So I will not remove any of them. Then we select an alignment algorithm. In this case, it's only mapped. And we create an alignment. When you do so, the system returns you to this window, and you see that the alignment is being calculated here. So let's create another one. In the meantime, this time an amino acid sequence, and I will choose also my hemoglobins, but now my amino acid sequences of all hemoglobins I have here in the system. So same thing, 23 different sequences, but in this case, those are amino acid sequences. In the first case, those were nucleotide. Same thing, we have duplicates. I will not remove any of them. And we have a new alignment being calculated. The previous one, the nucleotide alignment, we double click on it and we can see everything, all our sequences, all our hemoglobins, nucleotide sequences aligned. Here you can select, for instance, if you want to show features, and features are, in this case, are the translation and the name of the entire CDS. In this case, you can select if you want to see them or not. You can use base colors or not, and you can also, in the case, for instance, you have a chromatogram. You can also visualize the chromatogram here. In this alignment, I don't have any. So let's say we want to analyze the alignment. So here we have two parts. We have all the sequences here. You can scroll down or up, and you have this small window down where you can select different places of the alignment, and you can go to different places in the alignment. Another way to explore the alignment is to, for instance, find mismatches. And if you click on these arrows, you can go one by one through all the mismatches in these sequences. In this case, you have 71 mismatches. You can explore all of them. If you want to search for a particular feature or for a particular sequence, you can go and search for it here. So let's say I want to search for GGA. Okay, here's the first one. I have 8 different GGA sequences in my alignment. So you can go through all of them one by one. This is one way of doing it. The other thing you can do is to just compress or zoom out your alignment. In this case, there is here another button that is not available. I just remember this because I will go to another example where I will use it. So this is a nucleotide alignment. You can see all of it here, and you can explore it anywhere. But since I really prefer protein alignments, because for me they are more informative. Sorry, this one. So you can go to your protein alignment. In this case, it's amino acids. It's not nucleotides. The difference with the nucleotide alignment is that in amino acid alignments, Teselagen calculates also a consensus sequence, if it's possible to do so. So here on top, you have your consensus sequence. And also you can scroll down and see the sequences of all your hemoglobins, alpha hemoglobins in this case. You can also turn on or off different. For instance, you can use amino acid color schemes. These are like very typical amino acid coloring schemes or not. You can color amino acids by physical properties. For instance, hydrophobicity. In this case, the red ones are the most hydrophobic. Blue ones are the less hydrophobic, more hydrophilic. So you can color them using any of these other properties. You can also select serine and threonines. Usually you can identify labile sites. And labile sites are those that do not conserve really a specific amino acid. So for the consensus sequence, you have an X. You can have, let's say, any amino acid there. That's a labile position. And you can just quickly identify them here. And the other thing that's really interesting here is that you can plot the conservation for each position. So you have 100% conserved positions, and some others, like this labile position, is only almost 40% conservation. The other thing that I really like is that you can color for properties, and those are properties that are conserved in a position. So for instance, in this position, all amino acids are positive. In this other position, the labile one, the only conserved property is that they all are small amino acids. So that's even important. Even if you have a position that's not really conserved, you have a conserved property. So you can explore it here. And if you want to know what properties are those, these are the properties that we are calculating here. So you always have this as a reference. So at the end, you have here all your amino acid alignments. Same thing as in the nucleotide alignment. You can find mismatches and run through all of them one by one. You can search for any sequence or any feature, anything you have. But in this window, you also have this information window. So if I select a track, you can also have the length of that specific track. Here you have information about the specific track, but here you also display some information and some other calculated, such as the molecular weight or the isoelectric point. This is a calculation. Extinction coefficients and the number of mismatches for this specific track, and the amino acid frequencies you have in this track. You can also select not a given track, you can select a small part, and you have different calculations here with different changing calculations. So in Teselagen, you can perform these alignments. Let me go back and show you this one, because there is another trick you can use. Here you have an alignment of different PET28B variants, and they have differences. They have indels here, some of them. They have some mismatches in other places. So if we go here, we have some mismatches here, an indel, small indel here, and so on and so forth. But the thing is that if you see and if you know your sequences, let's say, okay, I'm showing here features and I have annotations here. So I know that all these sequences have a C-terminal 6-his tag. The one on top has this C-terminal 6-his tag in this part of the alignment, but all of the rest have the same 6-his terminal here in the beginning, here. So I know that something is not very good in the alignment because I would like this C-term 6-his tag of all the vectors aligned with the one on top. Since these are circular sequences, it's a little difficult. What can I do if I have something like this? I can just select this position here, and now I can use this button. I can re-index the alignment starting from this correct position. So if I do so, a new alignment is calculated, and this new alignment will contain all the different sequences, but now in the correct order. Every one of them is aligning the 6-his tag, the C-terminal one, in the same position. It's much ordered, and you can see that there is really indels and nothing else. Some others are shorter, but nothing else. So it's a cool thing you can do. And we are preparing also different things to enhance these tools in the future, in the very next future. So these are the sequence alignment tools in Teselagen. The other thing I wanted to show you is the PCR calculation tools. And for that, you start with a DNA sequence. So you go to your molecules in Teselagen, you select any DNA sequence. Let's say I wanted to use this. It's one of the variants of PET28B I have here. So if I go to the editor, I have all the sequence here with all the features and so on. So let's say I want to perform a PCR using this as a template, this plasmid, and I want to amplify this cannabicin resistant gene. I can select it any way, really. Sorry, there is a window here. Yeah. So you can select it any way. I can select the feature directly, and I select everything. I can go directly to the sequence and select anything I want, but just take advantage of the Teselagen things, and you can select the feature you want to amplify in this case. Once you have a selection, any selection here, for creating a PCR and primers for that PCR, you right-click on your selection, you select Create, and go to the last option here, which is Create PCR Primers for this region. Then this window opens. You have to give a name of your a name to your Amplicon. So this is KanR, the KanR gene. I will select the positive strands for creating the PCR primers. So the positive is the reference. You could select the negative if you want. And you could select tags to be associated with the Amplicon that you will create, or your primers. I will not select anything here. So this is important because all of them will be saved in your Teselagen library at the end. If you wanted to tag something and label something, you could do it just using this way. But the important thing is the parameters here. First thing, you choose what are your primer binding regions. You can choose any. That means the tool can design primers for any part of the sequence you have selected. You can select internal primers. That means the primers have to be internal to that sequence. It doesn't have to be the entire sequence, but internal to it. It could be smaller Amplicons. Plus, primers can be flanking, the region you selected, so you amplify externally, or you can design plasmid primers, that are overlapping the 5' and 3' ends of your selection. So you can choose any of them. The other parameters you can modify are in the case, for instance, you select any, or you select internal or flanking, you can have different product sizes. So you can select your minimum or maximum product size if you want to. If we select, for example, this option means that I want to amplify exactly the region I selected, not externally, not internally, but exactly that region. So the primers have to start at the beginning and end of that region. So here you don't have to select the minimum and maximum size because it's the size of the region you selected. Then you select your desired primer minimum and maximum temperatures, melting temperatures, the sizes of your primers, the ideal ones, the content of GC in your primers, the melting temperature for your allowed hairpins in your primers. If you want to introduce a GC clamp at the end, at the 3' end of your primers, you can specify it so. And here is how many identical nucleotides you allow in a row in your plasmid, in your primer, sorry. So let's say I want to amplify my selected region, and I use the default parameters. I click on OK, and I wait for the Teselagen to calculate. So I have some errors here. So there is a long polynucleotide sequence, GC content failed, and so on. So in this case, I can change different things. So if the GC content was so low, I can just use wider limits. Let's say I do the same for primary length, and since there was a very long sequence with identical nucleotides, I modify this one. We go again. Still, I have errors, high hairpin stability, for instance, low TMs, blah, blah, blah. So let's modify that to 40 and this one. Yes. So now with these limits, Teselagen created 2 different primer pairs for my amplification. Both pairs amplify the same product. So it's the exact same region as I selected. So it's this region. So the product TM, the Amplicon TM is just the same. Same region also on my selection. And I have these melting temperatures for my primers, only 3 degrees of difference, not a lot really, not perfect, but these are primers that have to align exactly at the 5' and 3' end. So not perfect, but perfect enough. Content, GC content is okay. It's a little low for the forward plasmid primer here, same for the second pair. But penalties are calculated and are lower in this case. If I wanted to choose one of them, I can choose both of them if I wanted. But let's choose just the first one because it's the one with lower penalties. Once you do so, the Teselagen calculates your forward and reverse primers. This is, this is a forward primer. Yeah, this is a forward primer, and it's annotated here, and the reverse primer is here. So both of them just start at the beginning of your selection. So it's exactly that selection. That was the thing I selected. And if you double click on any of them, you have the sequence of your primer. And here, if you wanted, you can modify the sequence. For instance, if I wanted to add a tag, a 5' tag to this primer, I could do so just by adding something, and it registers here in the alignment of the primer to your sequence. If I wanted to, let's say, I wanted to design a mutagenic primer, I can also change a base here, and that is also reflected here in your primer sequence. So let's say this was an A. I will just delete everything. So here you have the information about your primer, about the GC content, the TM, and so on, and you have the possibility of editing your primer if you want so. So here, after you have your primers, you can use another tool in Teselagen to simulate a PCR. So here I have selected my former kanamycin R primer, and I selected this. Oh, sorry, this. I lost my other primer. This is strange. Okay, this should not happen, and I'm sorry for it. I probably deleted it. So the idea is that you can just simulate your PCR. You choose 2 different primers you have on your sequence, and you can simulate the amplicon you have using this tool here. I'm sorry because probably I deleted my reverse primer here, but just to show you what kind of tools you can use, I introduce you to the PCR simulation tool. If you selected different kinds of primers, the primers should all be annotated on your sequence here. In this case, I just selected 1 primer pair, and it's the only one I have. Teselagen also registered a new part, which is the Amplicon. Your kanamycin R Amplicon is registered in Teselagen, and also you have the oligos for this amplification. The reverse and forward oligos are here. So they are already incorporated in your Teselagen library. If you wanted to, let's say, explore their sequences or explore where they are binding and so on, you can explore directly from the DNA sequence library. Okay, so this is the end of this demonstration. It's a very simple PCR design tool. The good thing is that, as we told you before, we are always developing new things, so this PCR tool will be enhanced. All the process of selecting new parameters, for example, now is being automated and suggested for you what the modifications could be, and if you accept them, that's automated, and you don't really need to think about what parameter to modify to get good primers for your amplification. So that's one of the things that we are thinking about, and one of the enhancements for the PCR design tool. Also, the sequence alignment tools are constantly being improved. So with this, I just will stop my demo. I just wanted to show you how these 2 tools are used and how they work. And I go back now to Jota, and we will continue with QA.
Thank you, Alvaro. Thank you for this presentation. Let's start with some Q&A. We have received some questions here. I'm gonna start with the first one. Probably we won't be able to go over all of them because we're almost on time. But don't worry, we're gonna answer all of them and send it over email. So how can I design primers for introducing mutations in an amplified sequence? Álvaro, Eduardo, if you can take that one. You are muted, Alvaro.
Always happens. So the question is,
Sorry again. How
can
can
you
I
repeat it?
design primers for introducing mutations in amplified sequence?
Okay, yeah, fine. I just showed you that you can edit your created primers. So when if you want to introduce mutations and you know which mutations you want to introduce, you can modify the sequence of your created primer just in the window I showed you. So I showed you how to add tags and how to modify internal bases in your primer. So that's a way. We are also in this moment developing a new tool that will help design mutagenic primers and introduce mutations in DNA sequences. And that will just be launched in the next few months, I think. So keep in contact.
Thank you, Alvaro. So while we share another question, I wanna leave our information. So everything that we're talking here is being recorded, and we're gonna upload it into YouTube and LinkedIn, and you can also reach out directly to us. So just have in mind that. That's because someone else was asking if we're gonna, like, if we could share this. Yeah, don't worry, we're gonna send this over email, share it on YouTube, and you can find it also on the landing page. Our last question will be, can I use existing primers for a PCR design?
Yes, I can take that. The
Okay.
answer is it's easy. We are just incorporating that as a new feature into the tool. In this moment, the tool only design primers for the sequence you select, and those are new primers. But we are developing just an enhancement to use primer oligo sequences you already have in your Teselagen library as a 5' or 3' primer for your PCR.
Okay, thank you, Alvaro. Yeah, that was an easy one and a quick one. So maybe we can answer a couple more. So how can I design primers for a Gibson assembly?
Okay, I can also take that. Okay, for Gibson assembly, this tool is a simple tool to design primers for a single PCR you want to perform in your lab. So you design the primers for that exclusively. If you wanted to design primers that are going to be used to amplify parts for a Gibson assembly, then you have to use our special tool for that. And let me share my screen just a little. Okay. For that, we have, in molecular biology, you have this tool. And in these tools, you have designs. You just combine different parts here. You define different bins. You define parts that are going to be incorporated into those bins. And in this case, we are testing different parts for the same construct. So here you select if you want to assemble this using, in this case, it's Golden Gate, but you can also select Gibson here. And if you select Gibson and you have selected also your parts and so on, when you submit this for assembly, the result of that will be a DNA assembly report. And let me show you one of them because there's the PCRs. Let me, Not this. Let me see this one. So here, as an answer, you have Teselagen has designed everything for your assembly, including the oligos you will use, and each oligo has overhangs that are introduced when you amplify each part you will use in your assemblies. So you have the oligos, you have the overhangs, you have the PCRs that you have to perform, and at the end you have all the assembly reactions. So everything is performed here. If you want to calculate primers for PCRs that will be going to be used in a Gibson assembly, then you use this tool. It's a more complicated one.
Yeah, I know we are on time, but just to complement Alvaro's response, I think what he just showed actually is what makes our DNA assembly planning algorithm one of the most popular tools in our platform because of its versatility and because it's known for making very good predictions about the likelihood of having your primers and your assembly reactions work. And it will also generate a number of warnings telling the user what might fail or not, and providing insights on how to adapt the assembly protocol. So yeah, it's a great tool also to explore eventually.
Great. Thank you, Álvaro, for this great demo. Eduardo also, and Mine for helping us have like this nice webinar with everyone here. Thank you everyone to that connected, and we're gonna be sharing this, as I mentioned before, over YouTube. We're gonna send the link to every participant here. So and please, if you wanna reach out or with more questions or anything, please let us know. The remaining questions that we didn't answer, we're gonna also share it over email. So thank you very much. I hope you have a great rest of your day. See you, everyone.
Thanks, Scott. Thanks, everyone.